colleagues. We will today continue a series of our lectures, of our webinars concerning on clinical part of pharmacovigilance, clinical pharmacovigilance. I receiving many interesting questions from you, and I'm very thankful for it. And one topic which now very actual, and I see what kind of question I'm receiving, it's drug induces liver injury. Why? Because so many factors now pluses and just increase this problem, drug induces liver injury, and many doctors asking me from overall additional information, and I decide to make this lecture. It's one long one, and after it I will make another lecture, and we will discuss real cases, patients which have drug induces liver injury, how was conducted management of this pathological condition. Let me introduce myself for my new participants. My name is Galina Cordero. I am qualified person pharmacovigilance, head of pharmacovigilance department, GVP.Фармак. I am clinical expert of GVP.Фармак 2. I am medical doctor, MD master degree, and with clinical experience working in hospitals and scientific experience too. I would like to tell that I will do everything possible, all best, that these lectures will be useful for practical medical doctors in daily practical activities, and because too many problems now they meet every day. Let's start. First of all, we will talk about hepatotoxic drugs. Medical doctors, pharmacists, pharmacovigilance specialists, pharmacovigilance safety physicians, and medical advisors and clinical trial specialists should know this group of drugs. We will talk about today in clinical trials too, about how to meet this situation very fast, because as situation have been shown that, for example, situation with valproate, yes, and it's another topic that it's I all time thinking about it, teratogenic effects in pregnancy, and how avoid situation in clinical trials when including and excluding criteria are present in clinical trials, how extrapolate these results in real population. Hepatotoxic drugs. Liver plays a key role in detoxifying harmful substances. Majority role, you know, it's system of cytochrome 450, and this role, this function, these enzymes cytochrome 450 play major role in detoxified substance too. Toxic hepatitis is liver inflammation due to toxic chemicals, drugs, or certain poisonous mushrooms can be too. Among patients with acute liver failure, drug-induced liver injury is the most common cause. Drug-induced hepatotoxicity or drug-induced liver injury, DILI, we will talk about shortly. DILI is an acute or chronic response to a natural or manufactured compound. Drug-induced liver injury, DILI, is a growing challenge because of the ever-increasing number of drugs used in medical care. After excluding acetaminophen, paracetamol overdose, you know what condition can be appear, and its condition called Reye syndrome, and very dangerous. Why it's actual? Because pandemia and too many cases of influenza different types became cause of rising administration of acetaminophen. And if you very often use or overdose, daily caused, but all other drugs can too, but after excluding it. Biological agents and HDS products is responsible for more than 10 of all cases of acute liver failure, posing a major clinical and regulatory challenge. Liver tox. Why we talk about liver tox? Because liver tox, and you see this reference, is in a clinical and research database developed by the National Institute of Diabetes and Digestive and Kidney Diseases, the National Library of Medicine, and the Drug-Induced Liver Injury Network DILIN study group to provide up-to-date, comprehensive clinical information on DILI. Liver tox includes an overview of medication, their chemical nature, indication, recommended dosage, and frequency of use. A description of the pattern and course of liver injury, case histories with laboratory data, and comprehensive list of reference. And after drugs that, according to analysis of data in this liver tox database, is classified on A, B, C, D groups, and we'll talk about every groups. Against in category A, we will talk about category A now, as a real potential hepatotoxins, and clinicians should be aware of that when evaluated the risk-benefit ratio of drug therapy. Treatment with these drugs should motivate physicians to guide patients about potential symptoms of liver injury when taking these drugs, and about prompt discontinuation if these symptoms occur. It means that should be very strict interaction and communication and dialogue between patient and physician who administer this drug for patients. All except one entity, estrogens, progestins, for example, or 98% had at least one convincing case that was associated with fatal outcome. It's a big problem. All of these drugs except telithromycin had been approved for marketing for more than 50 years, 33% for more than 35 years. The most common types of drugs were antimicrobials among 33% of the drugs, followed by drugs acting on the central nervous system, cardiovascular system, rheumatologic, antiaplastic, endocrine, and other types of drugs. Also, antimicrobials that were the most common agents among drugs. Antimicrobials were also the most common agent in categories B, C, and D. Antibiotics have been shown to be the dominated type of drug in both prospective and retrospective studies on DILI. This study you see reference, categorization of drug implicating causing liver injury, critical assessment based upon publishing case report, Hepatology 2015. Reference you see, plus you see reference liver tox. You can check it and use it in practical activity too, because as I said before, hepatotoxicity and drug-induced liver injury is very actual now, and it is a big problem concerning on risks appearing when you administer this drug. There is unfortunately not enough room to discuss many of these well-documented hepatotoxic agents. As mentioned in the abstract, azathioprine and, for example, infliximab have in one study been found to be associated with the higher risk on liver injury. And you see this to reference. Both hepatocellular and cholestatic injury has been described due to azathioprine. Despite the common problem of hepatotoxicity with azathioprine, there is a lack of studies with a significant number of well-characterized patients with this type of liver injury. I would like to demonstrate this list from Living Tox. You see it's 48 drugs. It's amiodarone, amoxicillin, clavonate, anabolic steroid, atorvastatin, azathioprine, busulfan, carbamazepine, chlorpromazine, contraceptives, diclofenac, diazepam, erythromycin, fluoxetine, gold stars. For example, if we were talking about amiodarone, it's interesting too because this drugs has two types of adverse reactions. From one side, it's hepatotoxic effects. A drug can be cause of development drug-induced injury. From another side, as you know, it's iodidated substance and medicines, and because of relations of iod in these drugs, it can be sulfur thyroid glands, and be very careful in patients with thyroid disease, and as you see, and it's hepatic, can be drug-induced liver injury. Why I pay you attention? Because you should have complex knowledge and look on it very carefully, because patient, as I said before, very seldom has just one disease. Plus, you see that one drug can be cause of several pathological conditions, and you should have very good clinical experience, clinical knowledge, clinical pharmacology knowledge, and I pay attention on this fact in every my lectures. If we're talking about pharmaceutical industry, the role medical clinical expert in company now is rising, because just clinical expert can rule it, can make activity with it, can solve these problems. It's why when, for example, QPPV and qualified person pharmacovigilance, the deputy QPPV, have no medical knowledge, medical education, clinical experience, you should have person with this knowledge, with this experience. We will continue our topic. We go ahead. It's another category B we will now talk about. Drugs in category B, data and liver tox. In comparison with category drugs, which almost exclusively had been associated with fatality, approximately 50% of category B drugs had been associated with a fatal outcome. It's a big percent, as you see. In drugs with less frequent reporting of liver injury in category B, only 38% had re-challenge reported, and 92 in category A, which suggests that the proof of hepatotoxicity is not there for all these drugs. You see this category B from Lift Tox. Drugs in category B, it's more than 20 and less than 4 cases that, according to analyze the data and liver tox, have been associated with more than 30 published cases of drug-induced liver injury. You see this list of drugs. It's amidoquinine, azithromycin, cyprotenon, heparin, notecan. Levofloxacin, oxofloxacin, interesting drug too because has several adverse reactions. Another one is influenced on tissue, for example. As you see, it can be even cause of some mitral valve injury or something like this. You be very careful with it. Imatinib, it's anti-plastic drugs and with enough big hepatotoxic effect too. Heparin is more often administered drugs and anticoagulant and has hepatotoxic effect too. It's why you should know these drugs and talk with patient and pay attention that hepatotoxic effect can be happen, adverse reaction can be happen. It's why they should be aware in it too. The top 5 implicated drugs in prospective studies daily. You see reference of these studies. It's studies drug-induced liver injury and analysis 461 incidents submitted to the Spanish register over 10 years period. And another one is causes, clinical features, outcomes of prospective study drug-induced liver injury in the United States. When we're talking about Spanish registry, what they pay attention in this registry, it's amoxicillin clavulanate. It's a very often administered antibiotics and enough good efficacy, but still don't forget. You should remember about this hepatotoxic effect. Isoniazid, it's anti-tuberculosis drugs. Diclofenac, it's NSAID drugs and steroid, very often administered too. And you see that flutamide, ibuprofen, and combination drugs can be caused too. Why you should know it? Because don't forget about drug-drug interaction too, and you should remember always that one patient has several diseases. Hepatotoxic drugs most often held responsible include anti-tuberculosis agents, other antibiotics, anesthetic drugs belonging to the holotan family. It's a very hepatotoxic drug, and non-steroidal anti-inflammatory drugs. Drugs withdrawn from the United States market since 1950 due to hepatotoxicity include pemaline, nefazodone, troglitazone, bromfenax, benaxaprofen, tylenic acid, and iproniazid. When we're talking about pemaline, Cillert was first marketed in 1975 for use in treatment of attention deficit hyperactivity disorder and narcolepsy. The FDA recommendation that the overall risk of hepatotoxicity associated with pemaline outweighed the potential benefit was withdrawn by its manufacturer in May 2005. Troglitazone Resolin was the first of new class of thiazolidinedione compounds. Peroxisome profilated activator receptor PRAR-G agonist approved by the FDA in January 1997 to modulate glucose level in patients with insulin-resistant diabetes. I can say that in this group, in this drug, we had big hopes that it can be very effective in treatment insulin-resistant diabetes. But at least 90 cases of liver failure, including 70 that resulted in liver transplantation or death, led to the withdrawal of troglitazone in March 2000. The decision to remove the drug from market was in part due to the approval of two new tislelidinedines, rosiglitazone Avanti in May 1999, and pioglitazone actors in July 1999. Despite adverse reaction can be cause of withdrawn, and we continue this topic. Withdrawn hepatotoxic drugs. Bromfenac Duract, its nesteroid inflammatory drugs, was launched in July 1997, a short-term analgesic for orthopedic pain. In June 1998, with less than a year of the market, it was withdrawn following more than 50 cases of severe liver injury, including 4 days and 8 liver transplants. Benoxaprofen Oraflex, another nesteroid anti-inflammatory drugs, first reached market in April 1982 and was subsequently removed in August of the same year due to the number of cases of fatal cholestatic jaundice. Thylaminic acid, tecrinafen, a diuretic drug developed to treat hypertension, was withdrawn in 1982 following reports of hepatic injury, including 25 deaths. Black box warning. Black box warning reserved limits a drug's usage by alerting physicians and pharmacists to the potential serious adverse effect of drugs. Recent drugs to have received a black box warning for hepatotoxicity include trovafloxacin, Trovan, tolcapone, Tasmar, felbamate, felbatol, and Xylitol, Ziflo. Challenges faced the prediction and prevention of DILI, drug-inducing liver injury. The case of troglitazone demonstrates the need to recognize signs of hepatotoxicity during drug development. During clinical trials, 12 of the 2,510 patients treated with troglitazone presented with alanine-aminotransferase ALT level of more than 10 times than upper limit on normal, and 5 had levels on more than 20 times comparing normal. Furthermore, a biopsy was performed in 2 patients, of which one developed jaundice, Watkins or Whitecomb, 1998. Also, no liver failure was attributed to troglitazone during clinical trials. These signs were predictive on liver injury, as evident once the drug reached market by report of severe and fatal liver injury in 24 of the nearly 2 million patients. And still, it can be a problem because not all, and we will talk about it, paramount challenges in clinical trial, because these results are difficult to extrapolate on real population. When we're talking about paramount challenges in clinical trials, I would like to pay your attention. Lack of specific diagnosis and markers or tests to verify episode of DILI. Limited cohort of clinical trials. The detection of merely 1 case of drug-induced liver injury, DILI, with an incidence of 1 in 10,000 would necessitate a clinical trial consisting of approximately 30,000 patients. Idiosyncratic nature of this reaction is challenged too, because while a universally accepted definition and characterization of such a reaction remained a point of contention, these types of reaction are unique to the patient. Lack of definitive correlation to the drug's known pharmacological effects, and vary in temporal patterns with regard to drug exposure. It can be potential relationship between the daily dose and prescription medication. Potential relationship between the daily dose of prescription medication and their propensity to cause serious hepatotoxicity. Examination of commonly prescribed oral medication in the United States in 2005 revealed as a significantly higher proportion of drugs with daily dose of 50 mg or greater were reported to cause liver failure and liver failure leading to liver transplantation or death than those with either dosage on 10 mg or less day or between 22 and 49 mg day. You see that there is potential relationship between daily dose, and if dose is more than 50 mg or greater, were reported a causal liver failure even. Drugs prescribed at or greater than 50 mg day counted for 77% of all serious drug-induced liver cases also. Additional studies required to confirm with initial observation these apparent relations between the doses of medication and DILI may assist in our understanding potential evasion of DILI during drug development patient usage. It's very important when, for example, stage 1, and you should pay attention as pharmaceutical industry on non-clinical investigation trials plus the first phase of clinical trials. First symptoms you can find there two first signs. Epidemiology of drug-induced liver injury. In the United States and worldwide, daily annual incidence in the general population under 50-20 per 2,000, 100,000. In population-based studies using different methodologies and cut off the crude annual incidence of daily post-marketing ranging from 2.4 to 13.9 per 100,000 inhabitants. In the United States, the most common cause of acute liver failure episode is 13–16%, yet it's important to know that it still is much less common etiology of acute liver injury overall. The incidence of idiosyncratic DILI is high in women. It's 59% when compared to men, which may be due to hormonal interaction with immunomodulated drugs or different pharmacokinetics. Patients more than 50 years old are more likely to develop DILI, possibly to increased prescription drug use. Acute DILI has been reported to occur in 5–10% of patients hospitalized for jaundice. You see a reference from source of epidemiology of drug-induced liver injury. Classification types of drug-induced hepatotoxic. There are several types, 3 types. It's intrinsic type of hepatotoxicity, idiosyncratic types of hepatotoxicity, and chronic types of hepatotoxicity. Hepatotoxic drugs from intrinsic DILI, drug-induced liver injury, it's acetaminophen, paracetamol, aspirin, tetracycline, and vitamin A. Definition: phenotypes and drugs associated with hepatic adverse reaction. Phenotypes of DILI, idiosyncratic DILI we now will talk about. Case definition: an adverse hepatic reaction that is unexpected on the basis of the pharmacological action of the drug administered. 3 patterns of DILI determined using of Ellis identified evaluation of liver enzymes levels. Initially, ALT activity, patient's ALT upper limit of normal of ALT and ALP activity, patient ALP is calculated. Then ALT is ratio, ALP ratio is determined. Hepatocellular patterns. If ALT alone is elevated more than 5-fold above upper limit of normal or ratio more than 5. Cholestatic patterns. ALP alone is elevated more than 2-fold above upper limit of normal, or R is ratio less than 2. Mixed pattern ratio more than 2 1 year after the onset. When we talked about medication associated with a phenotype, it's antimicrobials, amoxicillin, clavunate, erythromycin, fluoxetine, interferon, alpha interferon, isoniazid, ketoconazole, minocycline, nevirapine, nitrofurantoin, pyrazinamide, rifampicin, cotrimoxazole, and sulfanilamide. Central nervous system is carbazepine, chlorpromazine, dantrolene, halatan, phenacetin, and valproate. Valproate is said below. Besides of hepatotoxic effect, hepatotoxic adverse reaction, drug-induced liver development has teratogenic effect, and it's contraindicated for pregnant women. Cardiovascular: amiodarone, hydralazine, methyl-dopa, quinidine, statin, atorvastatin, and simvastatin, immunomodulator, azathioprine, 6-mercaptopurine, infliximab, interferon beta, methotrexate, and trioguanine. Antiaplastic: busulfan, fluoxuridine, and flutamide. Rheumatologic: allopurinol, auronafin, gold products, diclofenac, ibuprofen, nimesulide, and sulindac. And endocrine: anabolic androgenic steroid, estrogens, progestin, and propiondiuracil. Others: disulfiram and ticlopidine. Drug reaction with eosinophilia and systemic symptoms, DRESS syndrome. Its case definition: drug-induced hypertensity involving multiple organs with systemic manifestation. Medication associated with this phenotype: it's anticonvulsant, carbazepine, phenacetin, and phenobarbital, minocycline, allopurinol, abacavir, and neviparin. Drug-induced autoimmune hepatitis. Patient presented with acute DILI, drug-induced liver injury, with serological and or histological markers of idiopathic autoimmune hepatitis. These drugs: diclofenac, halatan, indomethacin, infliximab, methyl-dopa, minocycline, nitrofurantoin, and statins. Secondary sclerosing cholangitis. Patient presented with acute DILI with histological and or magnetic resonance cholangiopancreatography evidence similar to those of primary sclerosing cholangitis. It's amiodarone, atorvastatin, amoxicillin, clavunate, gabapentin, infliximab, 6-mercaptopurine, sevoflurane, and venlafaxine. Granulomatosis hepatitis. Case definition: presence on liver biopsy of granulomas, focal accumulation of modified macrophages that are attributed to exposure to one or medication. Allopurinol, carbazepine, methyl-dopa, phenacetin, quinidine, and sulfonamides. Acute fatty liver is phenotypes of DILI. Case definition: it's clinical syndrome of rapid development of liver and other organ failure associated with extensive microvesicular steatosis. It's amiodarone, dianazine, stavudine, valproate, and zalcitabine. Drug-associated fatty liver disease. It's non-alcoholic fatty liver disease attributable to exposure specific medication. Medication associated with this phenotype, it's methotrexate, it's 5-fluorouracil, irinotecan, tamoxifen, corticosteroids, lamivudine, and mifepristone. Nodular regenerative hyperplasia, case definition, diffuse nodularity with the liver with characteristic arrangement of hepatocytes and the center and periphery of nodule. Medication associated with this phenotype is azathioprine, busulfan, bleomycin, cyclophosphamide, chlorobenzil, cysteine, arabinozide, carmazine, daboxarubicin, 6-thioguanine, and oxaliptidine. Ductopenic vanishing bile duct syndrome, chronic cholestasis associated with bile duct loss, is azathioprine, androgens, amoxicillin clavulanate, carbazepine, chlorpromazine, erythromycin, estradiol, fluoxetine, phenytoin, terbinafine, and cotrimoxazole. Liver tumors, its characteristics of hepatocellular adenoma or carcinoma based on establishing histological computer tomography or magnetic resonance imaging features. Anabolic androgenic steroids and oral contraceptive as a medication associated with this phenotype. We will continue our topic. We will discuss risk factors for drug-induced liver injury. As you see, there are several risk factors for drug-induced liver injury: age and gender, genetic factors, dose and duration of drugs, kidney damage, alcohol and cigarette smoke, drug-drug interaction, diseases, HIV, hepatitis C, CRA, SLE, obesity, and nutritional status at least. Age. Age may be considered and contributing factors determining the susceptibility of DILI secondary to particular drugs and contributing to the phenotype of DILI. The susceptibility to DILI is greater in adults than in children, which may be a result of decreased clearance, reduced hepatic blood flow, and compound drug-drug interaction. Adults have an increased risk for hepatic injury from halotan, nitrofurantoin, fluoxetine, and in particular isoniazid, for which a few reactions have been reported in patients younger than 20 years of age. Aspirin and erythromycin acetylate, for which adverse hepatic reactions are predominant in children. You should remember it. Evidence extrapolation from level 2 studies, prospective cohort studies, and level 4 studies, case series. Example: adults have an increased risk for hepatic injury from halotan, nitrofurantoin, fluoxetine, and in particular isoniazid, for which few reactions have been reported in patients younger than 20 years of age. Patient risk factors DILI. Patient risk factors: the genetics, age, older or younger, gender, male, female, race, pregnancy, malnutrition, good microbiome, hormonal status, obesity, diabetes mellitus, comorbidities including pre-existing liver disease or HIV, and indication for therapy, for example, hepatitis C. Sex. Female sex may be considered a risk factor for DILI associated with specific drugs. Evidence level 4 case series: female sex may be associated with a greater risk of drug-induced injury. Evidence extrapolation from level 2B studies, retrospective cohort studies. Hepatoxic drugs often produce DILI with autoimmune features, and women are more susceptible to idiopathic liver injury. Evidence from several studies support from female patients with DILI may have a higher risk of progressive to ILF. Women are also more susceptible to hepatotoxicity from halatan, chlorpromazine, and diclofenac. Remember that diclofenac has, besides of hepatotoxic effect, very big, very bright. Its ulcerative effect, its ulcerative disease peptic acid can be happen in patient too. Risk factors for drug-induced liver injury. We will now talk about race and ethnicity. Ethnicity should be considered a risk factor for DILI. Evidence extrapolation from level 1 inception cohort studies. Example: trimethoprim, sulfamethoxazolone, methyl-dopa, and phenytoin were more often the cause of DILI among African Americans, while amoxicillin clavulanate was a causative agent in significantly higher proportion of Caucasians. Example 2. Slowonate 2 genotype was associated with increased risk of drug-induced liver injury among people of East Asian and Middle Eastern origins, but no association were found in Caucasians. Environmental factors DILI: it's smoking, alcohol consumption, infection, or inflammatory episodes. Regular alcohol intake may be a contributing factor for DILI associated with specific drugs such as isoniazid, methotrexate, and halatan. Anabolic steroids were found to be the most common cause of DILI among the heavy drinkers. Evidence level 4 case series. Risk factors for drug-induced liver injury. Drug-dependent risk factors include dose and hepatic drug metabolism, lipophilicity, concomitant drugs potential interaction, special chemical moieties, reactive metabolites and oxidative stress, mitochondrial hazards, hepatobiliary transport inhibitions. Comorbidity. Components of metabolic syndrome should be considered risk factors for the occurrence and the degree of adult in patients treated with tamoxifen and methotrexate. Evidence level 1B and 2B studies, RCT, and individual cohort studies. Chronic hepatitis B and C can be considered as risk factors for DILI from anti-HIV and anti-TBC tuberculosis therapy. Evidence level 2A study, systematic review of cohort study. Drug-dependent risk factors DILI: daily doses of more 50 to 100 milligrams of drugs with greater lipophilicity, stanol water partition coefficient more than 3, appear to more inducive with DILI compared with dose less 50 mg day or less lipophilic agents. Higher doses may lead to increased intrahepatic levels of the parent drug or metabolite. Greater lipophilicity may require more extensive metabolism, which can lead to covalently bound haptens that produce an adverse immune response in genetically susceptible individuals. Drug-induced autoimmune hepatitis occurs more commonly with drugs that undergo extensive hepatic metabolism. 90% of ILF cases occurred in patients receiving more than 50 mg per day. Further, drugs that are metabolized via cytochrome 2CD or cytochrome 2C19 appear to be associated with a greater risk of DILI than medication metabolized by cytochrome P3A or cytochrome P2D6. Mitochondrial disorder. A daily dose more than 100 mg, whatever the drug, predominant hepatic metabolism by cytochrome P450 enzymes, the formation of reactive metabolites, and dual inhibition of mitochondria and BSAP function are properties of drugs that can confer a risk for DILI. Predictive algorithms and selected preclinical tests are recommended to identify these liabilities in drug development. Evidence, extrapolation from 2C studies, outcomes research, and mechanistic studies. Mechanism of drug-induced liver injury, you see this is cascade. Include hepatic dysfunction, mitochondrial dysfunction, oxidative stress radicals, RTAP depletion, and as a result it can be hepatocyte death, apoptosis or necrosis. And you see that it can be it can be on stage 1, phase 1, when reactive intermediates, phase 2 can be connected with mitochondrial dysfunction, and can be reason besides on apoptosis and necrosis. It can be a reason of typical pathological process as inflammation, activated oxidative active radicals. It can be anti-inflammatory cytokines can be activated, interleukin 1, interleukin 6, and it can be cause of appearing antibodies too. We see that mechanism of drug-induced liver injury are metabolized by the liver P450 system in a series of phase 1 and phase 2 reactions. You see in left column. Toxic intermediates can elicit hepatocyte damage and death by induced apoptosis or necrosis. It's center column. Drugs that bind to cellular membranes can elicit an immunological reaction upon presentation to major histocompatibility complex MTAs particles, resulting in inflammation. You can see right column. Mechanism of drug-induced liver injury. The vast majority of drugs are liposoluble and metabolized in liver and excreted in bile or urine. The first step of drug metabolism is known as the phase 1 reaction and is mediated by enzymes of the hepatic cytochrome P450 system. Intermediate bioactive products generated in this step may interact with various cellular organelles, including mitochondria, leading to hepatocyte dysfunction and cellular demise. Potentially toxic intermediate products are then inactivated through glucuronaglutathione or sulfa conjugation, a subsequent phase 2 reaction. In order to limit hepatotoxicity, the generation rate for phase 1 products should not exceed the liver's capacity to inactivate them. Depletion or deficiency of the compounds responsible for phase 2 conjugation reaction may result in accumulation of toxic metabolites. Such is the case in patients who abuse alcohol and ingest acetaminophen. In this example, even low dose acetaminophen can result in several liver damage. Please pay attention on this fact. The inhibition of the mitochondrial respiratory chain, resulting in increased reactive oxygen species, radicals, and depletion of adenosine triphosphate. Several mechanisms contributing to mitochondrial dysfunction. The mitochondrial respiratory chain may be inhibited, diminishing and RTF production and resulted in increasing ROS levels. Amiodarone may inhibit oxidation of fatty acid, resulting in steatosis or steatohepatitis. Please, cardiologists, remember it too, and pharmaceutical industry who has these drugs in own register. Dideoxynucleotide analogs often used in the treatment of HIV may impair mitochondrial DNA replication. Drug toxicity may also result from the opening of the mitochondrial permeability transition for MTP, which is strongly associated with cell death. ROS generation, ATP depletion, and the aforementioned mitochondrial insults may combine to induce intracellular damage. Ultimately, hepatitis commits to apoptosis, but the process requires energy ATP which may not be available due to mitochondrial dysfunction and depleted ATP stores. In this instance, hepatocyte death occurs through the necrotic pathway which enhance hepatic inflammation. Immune-mediated injury. Immune-mediated injury is also an important mechanism of DILI and may be characterized by a prolonged interval between administration of drug and recognized liver toxicity. The liver contains components of both the innate and adaptive immune system. Bioactive drugs metabolites bind to cellular proteins and are exposed to major histocompatibility complex MMTase molecules on antigen-presenting cells. The interaction triggers an immune response directed against the hepatocytes. Halatan, for example, triggers the generation of antibodies directed against the tachrome P450, tachrome 2A1 source. Identified drug-induced antibody in patient blood may help in the diagnosis. Apart from antibody-mediated cell death locally, releasing cytokines and ROS also enhance hepatic injury. As classically described with halogenated anesthetic, immune-mediated DILI, drug-induced liver injury, may be much more pronounced and severe after repeated exposure to the medication. A careful medication history may reveal important information regarding reactions that appeared after previous administration of respective drugs. Pathophysiology of DILI. The intrinsic mechanism is both predictable and reproducible from drugs that are known to cause liver injury in a dose-dependent manner with a short latency period. For example, in acetaminophen toxicity, paracetamol, the metabolism of the drugs leads to the production of reactive metabolites that excessively accumulate, causing apoptosis and necrosis of hepatocytes. Example, acetaminophen toxicity as the intrinsic mechanism of DILI. For example, in acetaminophen toxicity, the metabolism of the drugs leads to the production of reactive metabolites that excessively accumulate, causing apoptosis and necrosis of the hepatocytes. These pathological processes can lead to acute liver failure and can be cause of death. It's why I pay attention, be very careful with dose and avoid overdose of acetaminophen, especially it can be very dangerous for kids. And please be careful with it and pay attention. Patients who consume acetaminophen at a single dose exceeding 7.5 grams experience acute liver toxicity, notable if plasma concentration exceeds 200 or 100 mg per liter or 8 hours after ingestion respectively. Acetaminophen intake at the licensed dose of 4 gram day over a period of 2 weeks results in elevation of alanine aminotransferase ALT above 3 more the upper limit of normal in 1/3 of patients. The idiosyncratic mechanism of DILI is another one. Include unpredictable course, unreproducible, independent of the drug dose. Pay your attention. Variable latency period generally beginning 7 to 14 days after first ingestion. Combination of host, drug, and environmental factors. Idiosyncratic DILI mechanism can be further divided into immune-mediated allergic liver injury from hypertensivity or non-immune-mediated metabolic non-allergic mechanism from mitochondrial injury. You see mechanistic relationship between intrinsic and idiosyncratic DILI. Drug include lipophilicity and dose characteristic, can be cause of reactive metabolites, it can be cause of hazards, covalent binders, glutathione depletion, stress kinase activation, mitochondrial stress, and reactive metabolites release, and stress at least, and it can be a cause for result of situation as lethal necrosis apoptosis, sublethal stress, co-stimulation. Sublethal stress can be lead to adaptive immune response, and adaptive immune response can lead to idiosyncratic DILI. Sublethal stress can lead to adaptive responsive and lethal necrosis apoptosis. It can lead to intrinsic drug liver induced injury. And you should remember there is relationship between intrinsic DILI, as you see, and idiosyncratic DILI, plus hazards, as you see, on level of mitochondria. Major factors can lead to genetic susceptibility. From one side, it can lead to intrinsic DILI. From another side, including genetic susceptibility, it can be lead to idiosyncratic DILI. It's why you should understand this mechanism at least. Hepatotoxic drugs classification. You see factors, you see comparative characteristic, intrinsic and idiosyncratic DILI. Factor incidence, more common is intrinsic DILI, idiosyncratic less, more common 1%. Predictability, intrinsic predictable, idiosyncratic unpredictable. Dose related, intrinsic dose dependent, idiosyncratic dose independent. Latency period, short latency period intrinsic, and idiosyncratic variable latency period weeks or months. Type of injury, intrinsic usually necrosis, and idiosyncratic is necrosis or apoptosis. Associated, intrinsic associated acute liver failure, idiosyncratic is rash, fever, and eosinophilia. Examples: intrinsic as a medicine is acetaminophen, idiosyncratic it can be isoniazid. Liver injury markers and significance when we're talking about hepatotoxic drug. Liver injury marker sorbitol dehydrogenase is hepatocyte-specific injury. Glutathione S transferase alpha centriolobular liver damage and kidney damage. HMB-GB1 high morbidity group box, it's necrosis marker. Glutamate dehydrogenases, it's mitochondrial disruption. Cytokeratin 18 fragments, marker of caspase cleaved proteins in apoptosis cell dish. M30, it's apoptosis marker. M65, total apoptosis and necrosis marker. Hepatoxic drugs, patterns of drug-induced liver. When we're talking about ALT, IST, when we're talking type of injury ALT as hepatocellular, it's more twofold rise, cholestatic normal, and mixed twofold rise. ALT normal, hepatocellular, twofold rise, more cholestatic, and mixed more twofold rise. ALT operation higher 5 is hepatocellular, cholestatic low to 2, and mixed from 2 to 5 ratio. Examples: hepatocellular is acetaminophen, amiodarone, allopurinol, HAART, and steroid anti-inflammatory drugs. Cholestatic is anabolic steroids, clopidogrel, chlorpromazine, erythromycin, hormonal contraception. And mixed, it's amitriptyline, enalapril, carbazepine, sulfanilamide, and phenytoin. Categories of patients with liability to cause idiosyncratic DILI. Tolerators are individuals who do not demonstrate abnormalities in liver tests while receiving the drug and do not develop any biochemical evidence of liver injury. IST or ALT values remain close to their baseline through treatment. Susceptible are patients who develop over liver injury while receiving the drug. They show progressive increase in aminotransferase that will continue while the patient is taking the drug. Patients may show fatigue, nausea, or vomiting, and may have right upper quadrant pain or tenderness. Decreased liver function will be accompanied by jaundice, elevated direct bilirubin, alcoholopathy, and may even progress to liver failure. Chronic DILI. DILI may be considered chronic when liver enzymes do not return to normal or pre-treatment baseline values after the drug is withdrawn and or signs and symptoms of liver disease persist 6 months after the onset of the drug-induced liver injury. A recent analysis from the Spanish DILI network suggests that unsolved liver test abnormalities 1 year after DILI onset may be more relevant to define chronic DILI, and you see a reference, you can use it in your practical activity. Categories of patients with liability to idiosyncratic DILI. Adapters exhibit transient elevators in serum IST or ALT that do not progress beyond the low-level state. Patients who adapt to a drug generally do not have symptoms or liver disease or decreases liver function such as jaundice, elevated direct bilirubin, prolonged prothrombin time, or increasing international normalized ratio. Transient elevated IST and ALT values may be quite common during the first few months of exposure to a new drug and do not predict DILI. While mild and temporary elevation of liver enzymes indicate mild liver DILI injury or injuries that spontaneously resolve, such changes are usually not clinically significant. Clinical symptoms of DILI. This type of hepatic damage, which is usually characterized by an initial early increase in ALT levels manifests itself as a cell death, characterized as either zonal or non-zonal in the form of necrosis and or drug-induced liver injury. Apoptosis, steatosis, and other types of hepatocyte degeneration. Drugs known or cause hepatocellular necrosis include sulfonamides, APAP, diclofenac, and pemaline. Submassive hepatocellular injury may be followed by hepatic fibrosis and cirrhosis, and examples of culprit drugs include methotrexate, isoniazid, and valproic acid. Clinical symptoms of daily cholestatic injury. Cholestatic injury is usually associated with an early elevation in alkaline phosphatase AP levels with or without hyperbilinemia and represents. The second most frequent type of DILI, its amoxicillin clavulanate, is one of the most common causes of acute cholestatic injury that resembles biliary obstruction. Anabolic steroids, methyltestosterone and fluoxamsterone, contraceptive steroids, and cyclosporine have been associated with poor cholestatic damage, and chlorpromazine and carbamazepine have been linked with cholestasis with hepatocellular injury. Clinical symptoms of DILI, mixed patterns of liver injury. A mixed pattern of liver injury is characterized by elevation of both ALT and ALP levels, and the clinical pathological manifestation includes those presenting both hepatocellular and cholestatic injury, as well as granulomatosis reaction. A mixed liver injury has been associated with carbamazepine, flutamide, phenobarbital, sulfonamide, and verapamil. The clinical symptoms are similar to other forms of hepatitis or cholestasis, where fatigue, nausea, malaise, pruritus, and jaundice predominate. In some circumstances, abdominal pain that is indistinguishable from acute cholestatis may be present. Concurrent with the theory that immunological mechanisms are responsible for certain forms of DILI, symptoms of systemic hypertensivity may be occasionally seen: fever, rash, and eosinophilia. Clinical features remarkably similar to primary biliary cirrhosis, prolonged jaundice, xanthomatosis, pruritus. While PBC may be result end-stage liver disease (SLD) and death, chronic cholestasis caused by medication is usually reversible and considered benign. Some forms of chronic medication-induced cholestasis are associated with destructive intrahepatic bile ducts. Types of drug-induced liver injury. Type hepatocellular. Enzymatic profile ALT MOSE2 upper level normal. Cerebral ALT ratio serum ALK and ratio MO5. It's more severe prognosis. Cholestatic ALT MOSE2 upper level normal, serum ALT, serum ALK, and ratio MO2. It's more prone to chronic disease, and mixed ALT MOSE2 upper level normal, serum ALT, serum ALK, F ratio between 2 and 5 more prone to chronic disease. Specific examples of DILI: acetaminophen accused dose-related DILI. A fraction of the drug is metabolized by cytochrome 2A1, cytochrome 1A2, and cytochrome 3A4 to a toxic intermediate metabolite N-acetyl-per-benzoquinine imine NAPOQ that can interact with intracellular proteins and induce hepatocyte death. Generated NAPQ is rapidly bound by glutathione DCT, which prevents the toxic effects. Hepatotoxicity occurs when glutathione is depleted or when NAPQI generation exceeds glutathione binding capacity. Glutathione depletion and increased generation and NAPQI occurs in alcoholics, and these patients can develop severe liver injury even with slow 2-4 gram per day doses of acetaminophen, paracetamol. Symptoms in the first 24 hours post-ingestion typically consist of nausea, vomiting, and malaise. Phase 1. The symptoms usually abate for 24 hours. Phase 2. Hepatocellular destruction occurs between 72 and 94 hours post-ingestion and is associated with abdominal pain and jaundice accompanied by nausea and vomiting. Coagulopathy, hepatic encephalopathy, and renal failure characterize several cases potentially resulting in death. Specific examples of drug-induced liver injury: anesthetics. Halatan-inducing drug-induced liver anesthetic usually occurs after multiple exposures and is thought to be driven by immunologic mechanisms. The conversion of halatan to 3-fluoroacetylchloride by cytochrome P450, especially by cytochrome 2A1, results in the formation of 3-fluoroacetylated proteins that serve as neoantigens and drive the production of autoantibodies anti-cytochrome 2A1 that mediate hepatic destruction. Clinical history may reveal fever and jaundice after previous administration. Hepatitis destruction is selected by elevated serum transaminase, while eosinophilia suggests the immune reaction. Biopsy findings may range from leukocyte infiltration to massive hepatic necrosis. More of the cases are mild, but acute liver failure may occur, potentially requiring liver transplant. Case reports exist. Liver toxicity from newer generation halogenated anesthetics such as isoflurane or sevoflurane is uncommon. Specific examples of daily anesthetics: acute dose-related anti-inflammatory drugs. Non-steroidal anti-inflammatory drugs are also an important cause of hepatotoxicity. Diclofenac, the most studied in the class, is glucuronilated and also subjected to cytochrome P450 mediated reaction that results in bioactive products. Both reactive metabolites and immune mechanisms mediate toxicity. Decreased prostaglandin synthesis due to cyclooxygenase inhibition may also enhance injury. Chronic diclofenac administration may result in elevated ALT levels in the first 4-6 months of therapy, but severe toxicity has also been reported. Bromfenac, nimesulide, and sulindac are the nonsteroid anti-inflammatory drugs and they most frequently associated with hepatotoxicity. Nimesulide administration has been reported to elicit severe toxicity resulted in acute liver failure. Sulindac and ibuprofen are associated with cholestatic DILI that is reversible after drug withdrawal, although fatal cases have also been reported. Specific examples of DILI antimicrobial drugs. Isoniazid is metabolized in the liver mainly to mono and diacetylhydrolysine and several other compounds. Genetic variation in rates of INH metabolism exist. Slow metabolizers are likely to develop higher transaminase levels in response to INC administration. Co-administration of drugs that increase cytochrome P450 activity has additional effect. Rifampin, for example, enhances the toxicity of INT. Most patients recover in several weeks after discontinuing the drug, while continuing the medication may result in severe hepatotoxicity, potentially leading to acute liver failure. Alone, seldom-induced DILI in up to 2.7 on patient. However, it may occur in patients with pre-existing liver disease. Rifampin holistically, rifampin completes with bilirubin for the bile salt export pump and results in hyperbilirubinemia and cholestatic liver disease. In addition, drug-inducing hypertensivity may also be responsible for a minority of cases. Pyrazinamide is generally not toxic per se, but when administered in combination with other drugs, INH, rifampin, ethambutol, or quinolones, the risk quinolones, the risk of hepatic adverse reaction is significantly increased. Therefore, rifampin is no longer combined with pyrazamide for treating latent tuberculosis infection. Acute dose relation DILI antibiotic drugs. Beta-lactams such as penicillin and cephalosporins are commonly associated with DILI. The presence of beta-lactamase inhibitor clavulanic acid significantly increases the frequency of adverse reaction leading to cholestasis or mixed patterns of liver injury. DILI induces but clavulanic acid compounds typically is manifested but reversibly jaundice, but several cases requiring liver transplant or resulting in fatal outcomes have been reported too. Petelinase-resistant penicillins are also associated with cholestatic forms of DILI. Macrolides are generally associated with reversible cholestatic liver injury too. The risk of erythromycin-inducing DILI is estimated at 3.6 on 100 cases, 1,000 cases. Patient present with abdominal pain, anorexia, nausea, and vomiting 2-4 weeks following the initial administration or after 2-3 days if re-challenging, suggesting a hypertensivity mechanism. Liver abnormality generally subside within 2-5 weeks after stopping the drug. In rare cases, cholestasis persists for up to 6 months. Specific examples of DILI sulfanilamide-induced liver injury. Sulfanilamide-induced liver injury occurs with the first month of administered the medication. Most forms of liver injury are cholestatic, but inflammation and necrosis may also occur. The patient usually recovers within several weeks after stopping the antibiotic, although chronic cholestasis or enhanced severity has been reported. Macrodantin is well recognized to induce both acute and chronic liver disease and may be indistinguishable from autoimmune hepatitis. Specific examples daily antifungal induced liver injury. Ketoconazole and other azoles are associated with an increased risk of hepatotoxicity. Liver injury generally present as increasing transaminase levels that are usually. The hepatitis patterns as most common cholestatic and mixed form have been observed. Patients on antifungal therapy require careful monitoring, and administration should be abruptly stopped if liver enzymes become elevated. Failure to do so can result in severe liver damage and death. Oral terbinafine rarely induced daily. However, several cases have been reported, thus monitoring liver enzymes at baseline and after 4-6 weeks of treatment may assess for the potential hepatotoxicity of the drug. Specific examples of daily lipid-lowering agents. Statin-induced irreversible dose-dependent rise in aminotransferase levels and very rarely result in liver failure. Monitoring the liver test at the beginning and during statin therapy is under debate. Autoimmune-like hepatitis has been reported in several cases and may be correlated with HLA DR3, DR4, or DR5. HCV infection did not significantly increase aminotransferase level during statin treatment. Ezetimibe, which inhibits the intestinal absorption of cholesterol, was initially reported to be safe. Ezetimibe was recently shown to induce cholestatic daily or an autoimmune-like hepatitis when employed alone or combination with simvastatin. Despite this association, statins can often be safely used in patients with chronic liver disease. Management of liver. The decision to discontinue medication is based on the values of liver enzymes. Drug administration should be stopped whenever ALT more 8 or upper limit of normal, ALT more 5 upper limit of normal for 3 weeks, ALT more 3 upper limit of normal plus bilirubin more 2 upper limit of normal. Prothrombin time international normalized ratio PT-INR more 1.5 upper limit of normal or in the presence of symptoms suggesting liver injury. Even after stopping the drugs, the outcome may vary from complete resolution to acute liver failure and death. With the exception of an acetylcysteine employed in acetaminophen intoxication, no other specific antidotes are currently employed. Criteria for liver transplantation. King's College criteria for liver transplantation: acetaminophen, drug-induced liver injury, and non-acetaminophen. pH less 7.3, it's for acetaminophen. Non-acetaminophen, INR more 6.5, ratio PT more 100 seconds or 3 of the following. Acetaminophen lactic acid more than 3.5 millimole at 8 hours. Non-acetaminophen INR 3.4 PT 50 seconds. You see creatinine, INR, and encephalopathy. And you see criteria: bilirubin, jaundice, age between 10 and 4, etiology, drug reaction unknown for non-acetaminophen. Please pay attention on it and please use it in practical cases because practical activity, because transplantation in can save life for patient, but it should be done in time and avoid death. Now we will stop first part of our lecture drug-induced liver injury, and from second part it be HIS law and how detected in clinical trials. Please, who interested in these topics, please follow to part 2. I will be very thankful for your question, for clinical question. Remember that after second 2 of these lectures, I will prepare second 3 where will be discussed real cases in patients with drug-induced liver injury, and for me it will be important your feedback as clinicians, because we together can be avoid and minimize risk of development of drug-induced liver injuries. Please subscribe on my channel, YouTube channel, leave comments, like my lectures, and I will be thankful for your needs, for your questions, and if you will ask many questions, we can organize, I repeat again, round table where we can discuss all acute questions, all important question, all actual question, because after, and I think this question will appear after third part of lectures. Now we will stop these lectures. See you on second part of lectures drug-induced liver injury. See you later. Thank you for your attention, my dear colleagues.